Okay, so everybody is attacking this by using Sark or RAS as a tool. But that's not how these oncogenes were first defined. The way these oncogenes were first defined is there was some tumor DNA, there was some mutation in a normal cellular gene that when mutated caused uncontrolled growth. That gene was isolated and defined an oncogene. So RAS came out of a mutation that caused uncontrolled growth. That mutation was found to be in RAS. So let's look at other tumors. Let's look at other viruses. Let's look at other tumors for cancer-causing oncogenes. Okay? And see if we always come up with Sark and RAS, or will we come up with the downstream components? And the answer is you come up with other components of the pathway. So the more tumor DNA you analyze and identify the cancer-causing gene, the more you identify additional genes in a common growth control pathway, right? By using cancer genetics, you can identify genes that are part of a normal growth control pathway that when mutated to become hyperactive, cause uncontrolled growth. Question? So she's suggesting that we look at what genes, so you're assuming that genes that are involved in this growth control pathway are all part of the same operon, or all controlled along the same piece of DNA. That would take another 20 years of advanced technology over where we were. And the answer to that is no, they're not. They're on different chromosomes, different locations. Okay, but it's good thinking. Okay. So what I have here listed are 3 additional components of this growth control pathway that were identified on the basis of them being oncogenes when mutated and activated. Okay? And they contain a normal cellular inactive counterpart, which then becomes mutated. And we determine their ordering with respect to one another in the same way that I just showed you we determine the ordering between Sark and RAS. If I introduce an activated form of RAS and an inactive form of one of these downstream effectors, then I will determine whether or not these effectors are downstream from RAS or not. If they're upstream, blocking them wouldn't affect RAS-mediated proliferation. If they're downstream, each one of the blocks of these effectors will block RAS function. If I use an activated form of this effector and block a downstream effector, I similarly order them with respect to one another. And these 3 genes are called RAF, MEK, and ERK, or MAP kinase. They are all protein kinases. So, yeah?
So if you block RAF and you see that MEK works, it still produces like uncontrolled growth, like wouldn't that, like how will you know that RAS is needed then for the pathway to go downstream? Because if you can just use MEK to get the uncontrolled growth, like what is the purpose of RAS?
So if you could use an oncogenic MEK to cause uncontrolled growth, what is the purpose of RAS? The purpose of RAS would then become in cells in which I don't have a mutant of MEK, in normal growth control. The only way MEK will be activated is through this whole pathway involving RAS. Okay? So the normal growth control pathway, where no mutations exist, lies in a tyrosine kinase upstream of RAS, and downstream of RAS, a kinase cascade, a cascade of 3 kinases, one activating on the next to activate the next. by phosphorylation. And this is what a growth control pathway uncovered using cancer genetics, a combination of cancer genetics and biochemical interactions, looking at the biochemical interactions between each of these members, uncovered this pathway and the specificity of how this pathway acts. And of course, now the question is, certainly we'd like to know what the downstream effectors of that kinase are. We'll talk about that. But now we'd like to direct our attention to get a complete picture of this control pathway, of the players in this pathway. We'd like to know how tyrosine kinase affects RAS activation. What is at the top of this pathway? Okay, is a tyrosine kinase, SRC or other tyrosine kinase, at the top of this pathway? What mediates normal growth control? What is the signal that initiates this pathway? And the answer to that came not from studies of cancer, but came from studies of normal growth control. And that was through the identification of the first growth factor. So people working in how cells normally grow, like how fibroblasts divide and then stop dividing, reach their morphological changes, were growing cells in culture and then adding blood serum in order to stimulate growth, because there were factors, in quotes, in blood serum which could stimulate cells to divide. And one of those people studying this was Stan Cohen at Vanderbilt University. And Stan Cohen identified the first cellular growth factor, which he called epidermal growth factor, because initially it was found to stimulate epidermal cells to divide. And he purified, sequenced, determined the structure of this growth factor. And this growth factor, again, is acting from outside the cell, and we're binding to receptors on the cell surface to stimulate growth. And the question arose whether these growth factors, the first of which was EGF, epidermal growth factor, but there were a number of others that were identified for different cell types. The question was whether or not these growth factors were using the RAS effector pathway to mediate, to stimulate mitogenesis. Were they part of the same pathway? And to answer that is the same way we've answered all the other ordering issues, and that is we know that if we add the growth factor to cells, they'll divide. If we add the growth factor to cells and block RAS function, will that block the growth factor-induced cell division? And the answer is yes. So the first link between the people doing growth control and cancer genetics came from the demonstration that downstream of growth factor action lied this growth control pathway uncovered by oncogenes and proto-oncogenes. What that would mean if one is a cancer geneticist is that, well, if that's true and the EGF receptor is upstream of RAS and RAF and MEK and MAP kinase, then we should find the EGF receptor as an oncogene. There should be human tumors or viruses that contain oncogenes